Today we'll be discussing Huntington's disease. This graph depicts the age of onset of Huntington's disease and the number of individuals who live and die in each category. The age of onset is highly variable, but the mode is between 30 and 40 years. It also shows how the earlier the age of onset, the more rapidly the disease progresses. In the United States, about 1 in every 10,000 individuals have Huntington's disease. Huntington's disease is autosomal dominant. Generally speaking, it is expressed in every generation. A child who has a parent who has Huntington's disease has a 50% chance of inheriting the disease.
Huntington's disease is a result of a CAG triplet repeat in a gene called Huntington, located on chromosome 4p16.3. In normal individuals, this triplet is repeated 10 to 28 times, but in people with Huntington's disease, it is repeated 36 to 120 times. The number of repeats correlates with age of onset. The triplet repeat codes for an abnormal stretch of amino acids called glutamines. This change causes a tighter bond to a protein called HAP1. This protein is involved in cell packaging and vesicle transport.
This defect in vesicle transport leads to localized neural cell death. It specifically targets and affects the nerve cells of the basal ganglia. This is a picture of a brain scan comparing a healthy individual to individuals with Huntington's disease. As you can see, a healthy individual has more brain activity, indicated by the red regions, than those affected with Huntington's.
Because of the damages to these areas in the brain, common symptoms include dementia, behavior changes, abnormal involuntary movements, difficulty swallowing, and speech impairment. Oftentimes, death is a result of these symptoms rather than complete neural degeneration. Currently, there is no definite cure for HD. However, a new field of study proposes the use and regulation of miRNA as a treatment for HD.